Now, please join me in welcoming Barry Gumbiner, Principal Investigator at Seattle Children's Research Institute's Center for Developmental Biology and Regenerative Medicine. He will be giving a talk titled, "What Cell Biological Insights into Cadherin Regulation Reveal About Disease Processes?" Thank you.
Thank you, and I thank the Allen Institute and all the organizers for giving me the opportunity to share our work with you. So I want to start from the point that cell-cell adhesion is typically a very dynamic process, even though one sees these static-like junctions in the electron microscope. And dynamics of adhesion underlie many important physiological processes. One that we've been interested in for many years is morphogenesis, where cells have to rearrange to cause tissues to change shape. Things like tumor cell metastasis involve changes in cell interactions. Inflammation, migration of neutrophils across epithelial monolayers, and of course cell signaling. So we're really very much interested in trying to understand how cadherin adhesion molecules regulate these dynamic adhesion processes. And there have been published many different kinds of models for how cadherins are regulated. Obviously, there's gene expression control. There's models proposed in which the cadherins are separated from the cytoskeleton, losing their ability to integrate tensile strengths. There's regulation by internalization. If the cadherins aren't present on the cell surface, they can't mediate adhesion. But we've been focused on this model here, where regulation of the homophilic bond occurs at the cell surface. And this is analogous to what many receptors do in different contexts, particularly integrins on platelets and leukocytes are regulated by this inside-out signaling. And I'm going to talk to you about our work studying this process. And just to illustrate why I think this is important compared to some of the other models, just sort of some cartoons here. On the left, the idea here is that while the cadherin-catenin complex links to the cytoskeleton, and I'll talk a little bit more about that, forming a junction. That is, if the adhesive bond itself is regulated by conformational changes in the interaction between proteins, that will allow junctions that are still interacting with the cytoskeleton the cytoskeleton can now exert force and do work on the cell junctions, in contrast to other models where they are internalized, so these junctions can be opened and closed by using the force. And just another kind of illustration, when you think about it, the adherence junctions are associated with an actin belt. If in the absence of a regulation of the adhesive bond, if this cytoskeletal exerts force and contracts, you might get bending of the epithelial sheet. But if the junctions are opened at the same time as a contraction, you might get opening and changes in the barrier or permeability properties of endothelium. So it's just sort of an oversimplified, but a way of thinking about the importance of this process. So we've been, for a number of years, looking at different kinds of models for understanding this process. We started doing it with developmental biology, but lately we've been using this model cell line called Colo 205s to help us understand this process, because these cells are chock full of cadherins and catenins at the cell surface, but they don't adhere to one another in culture unless you trigger them in some ways and activate adhesion. And one of the ways that we've been studying this process is to make activating antibodies, so antibodies that bind to the cadherin on the cell surface and trigger this adhesive process, so the cells convert from a leukocyte-like morphology to an epithelial sheet. And I won't show you the data for this, but we have several regions of the molecule. This is the crystal structure of the cadherin molecule, ectodomain, that seem to play a role and change, and some have activation epitopes associated with these changes in adhesion in these cells. So we have this model in which we think there's a structural change in the cadherin that underlies changes in the strength of the adhesive bond or the adhesion between cells. And as I said, there's evidence for changes in the extracellular domain, but we also think that these changes are controlled by the catenins and signaling process inside the cell, in particular P120 phosphorylation, which I won't talk about today. So I will tell you divide the talk into three different parts. One of our major effort in trying to understand the mechanism controlling this process is a structural biology approach I'll start off with. I'll tell you it's a work in progress. We don't have the answer yet, but we have some intriguing preliminary data. And then I'm going to talk about the roles of these changes in some pathophysiological processes, in particular two little stories, one on tumor metastasis and the other one on regulation of inflammatory disease, in fact, in particular, inflammatory bowel disease. So I'll tell you a little bit about the atomic structure of the cadherins. This is a crystal structure of the Xenopus C-cadherin ectodomain that we solved in collaboration with Larry Shapiro at Columbia, who's the crystallographer. And here's the different cadherin repeats and the calcium binding sites between them. A key interaction is between the so-called strand swap dimer, where a tryptophan in the second position inserts into a hydrophobic pocket on the other molecule, and that's a strand swap. And this is thought to mediate the actual adhesive bond. But another important dimer has been observed in crystal structures, especially when the tryptophan is mutated. It's the so-called X-dimer, which happens between the EC1 and EC2 domains in this region. And the idea here is this X-dimer is not the adhesive interaction, but is an intermediate along the path to forming this dimer. This has been published by several groups. And the idea here is the monomeric cadherin on the surface, the tryptophan tucks into its own pocket in an intermolecular interaction. Then it first forms this X-dimer intermediate, which then triggers catalytically the swapping of the tryptophans to create the strand exchange, and then the X-dimer dissociates and gives you the final bond. That's kind of the current model that's in the literature. So our first approach to trying to understand the nature of this structure is to try and get structures of our activating antibodies in complex with these cadherin molecules, because perhaps we'll see these activating antibodies causing some changes in the adhesive bond. So the first effort in a collaboration, we've gotten crystal structures of activating Fab fragments with this cadherin EC1-EC2 domain. And there's two different Fab fragments, and here's the structures shown. This is an activating antibody that binds mostly to the EC1 domain across from the W2 TRP2 residue in the EC1-2 domain. And this work is being done by Allison Maker and Leslie Ann Schechterson in the laboratory in collaboration with Bart Staker and Brad Hammerstein, who were at Center for Infectious Disease Research, which is now part of Seattle Children's. And this is a second Fab that activates adhesion in this model that I showed you. And it seems to bind EC2 and the interdomain region, the calcium binding site. Now, interestingly, we do see these antibodies causing some, or we see a small molecular change associated with these antibody binding. And Allison in this experiment basically overlaid the Fab cadherin structure with the already known cadherin domain structure. And in both cases, we see a little bit of a displacement of this TRP2 residue that's involved in forming the homophilic bond. In the second Fab fragment, the displacement's even larger. And you can see an expansion here of these structures overlaying all three. We see a slight shift in the TRP2 in the bond. Now, this is hardly proof that this is an important change in adhesion, but it generates a hypothesis that maybe these activating Fabs in some way enhance the formation of this TRP2 dimer. And we have several experiments underway to try and understand that. Now, it turns out Allison also overlaid these structures with this so-called X-dimer that's been published. And what's interesting about that is that the Fabs kind of clash with the potential formation of the X-dimer. In this case, this one FAB has a small clash, sort of emphasized here. In this structure, the second FAB completely clashes with the ability of the cadherin to form this X-dimer. And I must say, at this point, this is still a little bit of a puzzle for us. Why would interfering with the putative intermediate actually enhance adhesion? There are several different possibilities we're considering, is that maybe this really isn't an intermediate, or maybe the antibodies allow these molecules to bypass this intermediate. But that's something, again, that's underway. Now, another important approach we're taking in the lab is Allison's working with support the biochemistry department to try and understand the entire cadherin-catenin complex in the membrane environment and what the effects of the antibodies might be in this context. This is just kind of a virtual cartoon of the cadherins and catenins, which have all been solved individually by crystallography. And all I want to say here is that she's been able to reconstitute all these components of the cadherin complex inside membrane nanodiscs, and is really right now doing some cryo-EM microscopy. We don't have a lot of conclusions to make yet, but just to show you that she really is starting to get some images of this cadherin extracellular domain. These are class averages from the cryo-EM, and these are ones with the FABs bound, and you can see the FAB fragments on them. So far, we haven't seen much about the catenins in this complex, and we're trying to understand why that is. Anyway, this is very much work in progress. So I'm going to turn to this more about the physiological role of this regulation in pathophysiological processes, in particular, in this case, cancer metastasis. And the background to this is that it's been traditionally thought that loss of E-cadherin expression is important for tumor metastasis. Cells undergo an epithelial-mesenchymal transition, lose E-cadherin, that allows cells to dissociate from the primary tumor and to migrate to distant sites. But there's a problem with this hypothesis, although that may still be true, that there are plenty of distal metastases that still express E-cadherin. And there's also evidence that an EMT isn't always required for metastasis. So our hypothesis is not the presence or absence of the cadherin expression, but the regulation that we've been studying is important, that the idea is that under conditions of various kinds of environments, that the cadherin activity at the cell surface can be modulated, allowing the cells to move and undergo metastasis. So the experiment here is to treat mice bearing mammary tumors, E-cadherin positive tumors, with activating antibodies and ask whether that affects metastasis. And this is an experiment done by Tae-Young Na in the laboratory, and she has a poster here this afternoon, so you can see much more detail. I'm just giving you sort of a brief overview. And importantly, this is a MMTV.PYMT model. This is a genetically driven endogenous mammary tumor model. And what Tae-Young has observed is treating these mice with these activating antibodies significantly reduces the number of metastatic nodules. So that, and I should point out, all these experiments are done in parallel with a control antibody we call neutral antibodies that bind to the E-cadherin but don't activate adhesion. So it's not simply the binding of an antibody to the cadherin that makes the difference. Then one of the questions Tae-Young wanted to address is where in the metastatic cascade do these antibodies have an effect in reducing metastasis? And there, as you know, metastasis is a very complex, multi-step process. Many things are going on, but what we kind of divided this into two overall steps is how cells get from the primary tumor into the circulation, and the second step is from the circulation migration into distal tissues. And to do that, she did the experiment again, this time instead of using endogenous tumors, using metastatic cell lines that we can inject into the animal. And first, to study the first part of the cascade, she asked whether the antibodies would influence the formation of the accumulation of circulating tumor cells, And in fact, the antibodies do reduce the number of circulating tumor cells. In the second experiment, she asked whether antibodies had any effect on cells injected directly into the circulation by a tail vein injection. It's a classic metastasis model. And again, she saw the antibodies inhibited the metastasis of cells from the tail vein injection. This suggests that the activity state of E-cadherin is involved in multiple steps in the metastatic cascade, and we're trying to understand this in somewhat more depth. And just really briefly to remind you that historically people thought metastasis was due to the EMT and the migration of individual cells. But in fact, it's well known now that it's really a problem of collective cell migration, very similar to morphogenesis. And we think by activating adhesion and holding cells in their high affinity adhesive state interferes in some ways with some of these steps in metastasis. Okay, so I'll turn to the last part of the talk, is thinking about the role of epithelial junctions in barrier function in epithelial tissues. And again, this idea that I think it's particularly relevant to this question is that the junctions can be open and closed under conditions that regulate migration of leukocytes, for example, and things like dendritic cell incorporation into epithelial tissues. So we know that there's many cases where the permeability barrier of the epithelium is regulated during inflammation. And this is work done by Sri, who's got a poster here as well today, and she'll show you this data in much more detail. And this is just an in vitro experiment with some airway epithelial cells. And these cells have been infected with respiratory syncytial virus. The reason for doing this model is it's an in vitro model of a problem with infection and barrier function. And it's been shown that RSV causes a decrease in transepithelial resistance due to the increase in cell monolayer permeability. And what she found is that these activating antibodies prevented the loss in transepithelial resistance caused by this RSV infection. But more interestingly is to actually look in vivo during a real inflammatory process. And we've been studying inflammatory bowel disease, in particular because the gastrointestinal barrier function has been implicated as very important. Obviously, the intestinal epithelium separates the whole microbiome from the immune system on the inside. And there's evidence that increased permeability associated with IBD precedes overt disease. And there's actually a familial association of permeability with families having inflammatory bowel disease. And E-cadherin gene in particular has been found in genome-wide association studies to be associated with inflammatory bowel disease, colitis in particular. So the hypothesis is that modulation of the barrier function is controlled by the activity state of E-cadherin. That's sort of the theme that I've been pushing today. And the IBD-associated inflammatory processes downregulates adhesive activity, and then if we activate adhesive activity with these antibodies, we might be able to enhance barrier function and perturb the inflammatory process. And these are and one model that Sri has been undertaking many different models of IBD, but this is the so-called adoptive T cell transfer model that immunologists really like as a way to induce, you enrich in a population of these CD45RB high cells. And when you inject those cells into animals, they develop inflammatory bowel disease. And these are some of the gross measures. Those cells cause weight loss over time because with inflammation, the animals lose weight. The green is the control cells that are not inflammatory. And the red is with the neutral antibodies, and the blue is when she treats with the activating antibodies. And so you can see in this experiment that the activating antibodies did reduce weight loss. And this is just the long-term effect. Another overt measure of inflammatory bowel disease is colonic length in this model. That for reasons that aren't well understood, the colon shortens during inflammatory bowel disease, and again, the activating antibodies rescue that. Importantly, almost done, in collaboration with a company called Histopathology, they did a blind analysis of the histology, and I won't go through the pictures, but just to point out that in all the various measures of inflammation associated with inflammatory bowel disease, mucosal thickening, inflammation score, gland, edema, and neutrophil invasion, we see that the activating antibodies inhibit significantly the inflammation caused by this adoptive T cell transfer model, reducing the sum colitis score. So in conclusion of this part of the talk, we think that enhancing the activating antibodies by enhancing E-cadherin adhesion enhances barrier function and reduces symptoms of inflammation. So I just want to thank everybody who's been involved. I mentioned this is Seattle Children's Research Institute downtown. That's our new building next door. Allison is a graduate student in the biochemistry department, and with help of the faculty there, she's been doing both the crystallography and the cryo-EM in collaboration with Bart. I did mention these people and their involvement. Leslie Ann Schechterson's been involved with many parts of this project because she's the antibody expert in the lab. And the IBD project is being pursued in collaboration with Takeda Pharmaceuticals in San Diego. Thank you.
Okay, thank you, Barry. We have time for just a few questions. If you could please raise your hand and we'll pass you a microphone.
I get the Rick's question.
Do you have any way of reducing activations so you can turn it on? Can you turn it off as well?
I guess the interesting question there is why. I think in most epithelial cells, you imagine the adhesion's pretty high. These colo-205 cells are a little bit unusual in that they have cadherins in this inactive state. It'd be nice to know what that is. We think in most cases in normal epithelial cells, adhesion is often reduced by growth factors that induce morphogenesis or cell migration and things like that. So, and that probably happens through P120 phosphorylation. Well, there are a paper we published several years ago. There are some E-cadherin mutations that are found in human diffuse gastric cancer, and we found that some of those mutations did block activation.
Okay, thank you again, Barry.